Bismillahirrahmanirrahim. Assalamualaikum semua. So for today's activities, we are going to discuss about the factors affecting enzyme activities, which are consist of the temperature, pH, substrate concentrations, and enzyme concentration. For the first factor that will affect the rate of enzyme is temperature, right? And so the chemical bond in enzyme can be easily transformed of the results of chemical changes and physical condition. So, how the temperature will affect the rate of enzyme reaction. So, regarding to the diagram or to the graph, the first phase is show when at low temperature, the rate of enzyme catalyzed by enzyme is very low. After that, when the temperature rises, so what will happen here is the kinetic energy of the substrate and enzyme also increase. How this work? It is work when the frequency of active collision between the substrate molecule and enzyme molecules increases. So when this happens, the rate of reaction between enzyme and substrate increase. So you must know every 10 degrees Celsius and every increases of 10 degrees Celsius, the rate of reaction controlled by the enzyme will double until it reach at the optimal temperature. So that is for the first phase. Second phase, regarding you can see for the number 2 there, which is that is the optimal temperature, which is the enzyme reaction is at maximum rate. So basically, the enzyme reaction is will work best at the optimal temperature around 37 degrees Celsius, which is our human body temperature. And then the third one is for the third phase. You can refer to the graph. What happened at the phase 3, which is after reaching the optimal temperature. So any further increases in the temperature means if higher than 37 degrees Celsius, it can be 50 degrees Celsius. So it will reduce the enzyme activity and stop completely at 60 degrees Celsius. So what will happen here is during this temperature, enzyme become denatured. So you must know what does it mean by it means by the denature, which is the chemical bond in the enzyme molecule will break at extremely temperature. So at this time, the enzyme are unable to retain the 3D shape. No more, the shape will be changed. So the active sites of the enzyme also change. So this will cause that substrate does not complement the active sites of the enzyme. Next, we will discuss about another factor that will affect the rate of enzyme, which is the pH. So the enzyme will react most effectively at their optimal pH, which is in the range between 6 and 8. As example, salivary amylase, it will work best at pH 6.8. But for another two enzymes that we learned that have a special or assumption here is for the pepsin, which is this pepsin is in the stomach. So it will best optimally work at the pH 1.5 until 2.5. This is because the hydrochloric acid inside the stomach need the acidic condition. Meanwhile, another one is trypsin, which is have in the duodenum. So the pH, the best pH here is 8.5. As we know, we are still discussing on the pH factor, which is once the pH change, it will change the ion charge at the active site and the substrate surface. So as a result, the enzyme substrate complex cannot be formed. But then, if the environment of pH returns back to the optimum level, means back to normal, so the charge on the active site will be restored. Means it become normal back. So the ion charge will be restored. Dia akan balik kepada asal. So the enzyme will return to their function as normal. But for certain case, which is extreme change in the pH value, Maksudnya dalam keadaan pH yang berubah secara ekstrim, sangat besar. So this will break the structural chemistry bond. Ikatan kimia, structure 3D tadi, dia akan terus ubah the structure of active sites of the enzyme. So at this time, dia akan panggil sebagai denatured enzyme. So how to spell denatured? D-E-N-A-T-U-R-E-D. Denatured means the structure of enzyme will be completely destroyed. Next, we will discuss about the substrate concentration. The higher the substrate concentration, the higher the rate of enzyme reaction. Refer to the diagram or the graph. The phase 1 shows that as the substrate concentration increase, so the rate of enzyme reactions also increase. This is because why? This is because the effective collision between the substrate and enzyme molecule also increases. But the reaction rate continues to increase at one time until reach the maximum level. That is the starting point at the phase 2. Next, we refer to the phase 2, which is that is the maximum level when the concentrations of enzyme become a limiting factor. Dalam bahasa Melayu, kita panggil dia sebagai faktor penghad. This is because all the active site of enzyme are saturated. Bermaksud, semua active site pada enzyme telah digunakan sepenuhnya. So, at this time, digunakan untuk apa? Dia digunakan untuk catalytic reaction. So, once reach this level, so the rate of enzyme reaction become constant. The last factor that we will discuss is enzyme concentration. Refer to the phase 1 in the graph. Shows that when the concentrations of enzyme increase, so the rate of enzyme reaction also will increase. This is because why? Because at the enzyme it have active site. So more active site there and ready for catalytic action. The second phase shows that if the concentrations of enzyme in the reaction double, means let's say from the concentrations of 20, it will double into 40. So the amount of substrates converted to product per unit time also double. But with one condition, which is there is no excess supply of substrate. Means kita hanya tambahkan kepekatan enzyme dan kita tidak ubah bilangan substrate. So di sini dia akan jadi meningkat. For the third phase, shows the constant level. This is because reach the maximum rate. The concentrations of the substrates become the limiting factor. So the rate of enzyme reaction can only be increases by adding more substrate. Means dah habis dah substrate yang nak ditindakbalaskan. Jadi kalau nak tambah lagi reaction tersebut, kita kena tambah substrate lagi. Sebab enzyme memang tidak dimusnahkan selepas reaction. Still there. Tapi bila dia nak buat lagi dah tak boleh. Sebab dah tak ada substrate.